Just going to be going over the SACE Stage 2 Biology outline and Topic 1 course for you guys. I've made a few videos just to kind of help you navigate your way through what's actually quite a challenging subject with a lot of content to learn. Now, just a disclaimer, I am not a teacher. I've never taught biology before. As I said, I'm a medical student, but I did achieve 6 merits in the SACE, including 1 in biology. So I think that I've learnt enough to be able to give you some advice and some tips and to be able to isolate really what's the most important parts of the course to take away. So I've made these series of videos to kind of help you navigate your way through some Stage 2 subjects, as the teaching and curriculums are often really vague and confusing. This was a problem that I faced a lot, especially in biology. What I was being taught was vague and I never understood what I was supposed to learn, what I needed to learn, and what could possibly be tested. Now, if you don't like these videos, then don't use them. It's as simple as that. I'm just making them to try and help you guys by giving you the opportunity to learn what's necessary and what's not. So topic 1 kind of starts off with this discussion of DNA and its structure, its function, and how it replicates itself. Now, this is really fundamental because the idea of DNA structure, function, and replication comes up again and again throughout Stage 2 Bio. So I would recommend learning this really well because things like DNA replication will come up again with things like transcription or with DNA-DNA hybridization later in topic 1 and topic 4. So DNA is deoxyribonucleic acid. You don't need to know what that means, but you do need to know that DNA stores and transmits genetic information. So DNA is basically universal, meaning that it functions the same way in all living things, and it has the same basic kind of structure and function in every single organism that's living. So DNA is a helical structure, which is this spiral that you normally see, and it's double-stranded. These 2 strands are called polynucleotide strands, and we'll learn a little bit about what that means later. The polynucleotide strands are held together by weak hydrogen bonding, which is basically a form of intermolecular force that occurs when hydrogen is covalently bonded to either nitrogen, oxygen, or fluorine. Now, in chemistry, in Stage 2 Chem, we learned that hydrogen bonding is really strong, and that's because of its relative strength compared to other intermolecular forces. But in bio, however, we think of hydrogen bonding as really weak because it's broken down quite easily in a bunch of different active processes that occur in the nucleus. So we're looking here at DNA nucleotides, and they're really like the basic unit of life in DNA. So polynucleotide strands are polymers and are composed of many of these monomers called nucleotides. Now, if you're not familiar with what a polymer is, it's basically a bunch of things that are joined together to form this kind of strand, and the polynucleotides are no different. So the monomers are kind of these building blocks. Think of them almost as like Legos that you assemble together to make a larger structure. And a nucleotide in DNA has 3 basic components: a phosphate group, a deoxyribose sugar, and a nitrogenous base. Now, these 3 parts, they're shown on screen. You don't need to know how to draw their chemical structure. You'll never be asked that. But you might need to draw this using simplified diagrams. So you might use a circle to represent a phosphate, a pentagon to represent a sugar, and then a square to represent the base. That's just one way. There are multiple ways of doing it. There are 4 main nitrogenous bases, being adenine, thymine, guanine, and cytosine, and these 4 bases make up every nucleotide that you can possibly have in DNA. So DNA molecules will never have other nitrogenous bases apart from these 4. So the polynucleotide strands of DNA are held together by weak hydrogen bonding. So hydrogen bonds are formed between these complementary bases, and each of the 4 bases can only pair with one other base. So that's this concept of complementary base pairing. So adenine will only pair with thymine, and thymine will only pair with adenine. Similarly, guanine will only pair with cytosine, and cytosine only pairs with guanine. So there are these pairs, and bases can only pair up in these particular pairs. It's quite simple when you think about it, but it's just due to a complementary chemical structure of the molecules. Now, you don't need to know these structures at all. You just need to know which bases pair with which other bases. So a common question is, why is complementary base pairing important? And the answer to that is very simple. It keeps the DNA organized, and it allows for hydrogen bonds to be broken easily during DNA replication. Remember, hydrogen bonds are weak in biology. So the arrangement of DNA kind of depends on what type of cell it's in. And there are two main cell types that we talk about in stage 2 bio, which is eukaryotes and prokaryotes. So DNA in eukaryotes takes the form of linear chromosomes, which are bound to these globular proteins called histones. Now, we don't really cover the types of proteins in terms of globular and fibrous and things like that, but you basically need to know globular proteins are just round. They're kind of spherical in shape. So these histones are positively charged, and DNA is negatively charged, and that's what creates this attraction that allows DNA to bind to these histones. This is really important a little bit later on when we look at histone modification. But just for now, just know DNA and histones have opposite charges, so attract. And DNA in eukaryotes is located in the nucleus of the cell, and we'll learn more about the cell organelles and different parts of the cell a little bit later in topic 2. So DNA in prokaryotes, however, is in the form of a single circular chromosome, like the one shown on screen. So it's very simple, it's not very complex to understand. DNA in prokaryotes is not bound to histones. That's just another feature of prokaryotic DNA. And another thing to remember is that prokaryotes don't actually have membrane-bound organelles. That includes the nucleus. So if there's no nucleus in a prokaryotic cell, the DNA obviously can't be found in the nucleus, and that's why DNA in prokaryotes is located in the cytosol. The mitochondria and chloroplast, which are other organelles in eukaryotic cells, they also have circular unbound DNA that kind of resembles what's in a prokaryotic cell. And we cover this a lot in topic 4, so it's something to keep in the back of your head. So just remember, mitochondria, chloroplasts, and prokaryotic cells all have the same DNA. So you might have covered DNA replication in year 10, and essentially it's the process by which DNA molecules can duplicate themselves, and they just replicate to form two genetically identical versions of one molecule. It's a 4-step process, and you need to know these steps because it's a very common 3 or 4 mark question. Describe DNA replication, or describe the process that occurs in the nucleus that allows DNA to replicate itself. And some things that you do need to know is that an enzyme, so it's called helicase, breaks the hydrogen bonding that's holding together the DNA nucleotides. And this exposes those nucleotide bases and allows them to be, participating in complementary base pairings with other new bases. So then, free nucleotides anneal to both separated strands. So anneal basically means they join, and this creates 2 new DNA strands. Then, another enzyme, known as DNA polymerase, creates a chemical bond between adjacent phosphate and sugar molecules. So this is called the phosphate-sugar backbone in biology, and it's really essential to giving DNA its structure and creating that double helical shape. Now, it's really important then that both new strands are elongated, so more and more bases are continually added, and this will continue until the whole molecule has had new synthesized strands. And this will create 2 identical daughter DNA molecules from one original molecule. Now, I went over that really quickly, so it's important that you take the time to pause if you need to, write down these steps, and try and understand each step and why it's necessary. It's not worth it me just repeating the slide again and again and again, because the only way to learn is to try and commit this to memory. But 100% I recommend learning these steps in a lot of depth, because you're 100% going to be asked something like this in a test or in an exam. So DNA replication is considered semi-conservative, and what that basically means is that half of the original molecule is conserved. So that's why it's conservative, and that's why it's semi. So each parent strand acts as a template for the synthesis of a new daughter strand. And as a template, it's again due to this complementary base pairing. If one strand has a certain base, only nucleotides of the complementary base can anneal along that strand and therefore be produced in the newly synthesized strand of DNA. So the result of replication, therefore, is two new DNA molecules that both consist of one original parent strand and one newly synthesized daughter strand. Now this is really important because semi-conservative replication does come up quite a lot. Now that's really all that there is for topic 1.1. And yeah, if you got a lot out of this video, make sure to let me know. And if there's things that you want me to cover in more depth, I'm more than happy to. I'm going through quite fast with these because I'm trying to give the most direct communication of concepts without a lot of this waffle that's in the background. Because I know that a lot of students complain that their teachers talk too much about irrelevant things. So I'm only covering what's the content that's necessary for your tests, for your exams, and for your learning as a whole. So if you really want in-depth explanations of everything, you're in the wrong place. I'm here to communicate the essential information. Thanks guys for joining in. I look forward to teaching the next lesson.